as well. I'm lucky enough to have excellent Wi-Fi here, so I don't suffer from that anymore. Welcome everyone, absolutely fantastic for our first of a 5 workshop series. We are really excited about this topic. The reason we're doing this expert series really is because all of you, you kept on asking questions about functional testing and pairing with genetic testing. So we thought, how fantastic, let's deep dive into it. We have access to the most brilliant speakers, as you well know and you've seen. So Annalie and Nikki have been working extremely hard to put together this incredible series, obviously with Candace hosting it on the platform, and today is our first day. So I would like to welcome Dr. Wayne Sodano. I'm going to tell you a little bit about him and how lucky we are to have him join today, and then we're actually going to hand over to Dr. Sodano. So let's get going. Right, so let me find my notes. This is one of those times where you don't want to wing it. You actually want to remember all the many things that Dr. Sodano has done. So Dr. Sodano is actually a chiropractic physician. He is also a diplomat of the American Board of Chiropractic Internists, a diplomat of the American Clinical Board of Nutrition, a certified functional and integrative medicine practitioner, and is also board certified in traditional naturopathy. In 2005, Dr. Sodano shifted his focus much more to teaching for the American Board of Chiropractic Internists, and this really began his journey in research and development, and especially in the areas of integrative and functional medicine. He is the author of a very excellent book, An Approach to Basic and Advanced Laboratory Interpretation, which I know Annalie is very dedicated to and uses a lot, as well as the integrative medicine approach to thyroid disorders. Dr. Sodano frequently lectures worldwide on various topics of integrative and functional medicine, and has been the director of clinical support and education for Vexia Diagnostics since 2017. And he provides clinical education in the form of live lectures, live and recorded webinars, but also provides a one-on-one clinical support to all Vexia practitioners. So I think that gives you a sense of his deep and wide appreciation, understanding, expertise and knowledge in the area of functional testing. So thank you for Dr. Sodano for joining us. I know that Annalie and Nikki have had a little bit of an insight in what we're going to be hearing today, but for the rest of us, we're very excited to get started.
Thank you so much. You know, I want to let the folks know that you know, I was also a 7th grade science teacher, so I get a little teachy, and you're many years ago, so it's a lot of fun to be able to do that and present it this way. So I need control over here so I can put the slides up.
You should be able to, Dr. Sodano. You're welcome to go ahead and share.
Just have to click share screen at the bottom where it's a little green.
I have ensured that it is multiple participants, so you're welcome to go ahead and there we go.
There we go. There we go.
You are on mute though.
Okay.
There we go.
Can everybody see this?
Yes.
Can everybody hear me? Let's start with that.
We can.
Yes, we can.
All right, that's great. Wow, just someone had just jumped on, an old friend of mine from way back, Dr. Riha from Houston. So I'm so glad you're here, Doc, and hopefully anybody else that I've interacted with. So I'm honored to be a presenter for this. This is an amazing thing. It's a topic that I like, and for those of you that don't know, I'm very inspired by 3x4 genetics, what they do. I think it makes a lot of sense when I learned about this a year and a half ago or so. So let's go ahead and get started. Obviously, you can see that this is my topic, functional laboratory testing. The goal here is to have a lot of fun. I don't mind being interrupted, not interrupted, but if you have a question, you need to fire something in. Hopefully it's not a lot. We can save them towards the end, but this is interactive, so I want to have a lot of fun with this. So I hope you all can get a cup of tea or get a cup of coffee, which I'm not opposed to because it has cholinergic acid in it, which can help break down biofilm. Isn't that interesting? So here's the course outline. I divided up into 8 parts, as you see. We're going to talk about some basic laboratory testing, factors affecting lab tests. Then I'm going to talk about biotin. Hopefully you all know about this in biotin interference. I want to talk about nutrients for proper red blood cell production. I want to talk about the Neutrophil lymphocyte ratio, which is something you can pull right off of a CBC, which is kind of cool, and get an idea for that as well. And it actually, I'll give you a little carrot here. You can use this almost as a pre-screen for COVID on who's going to potentially get worse if they happen to get COVID, which is kind of interesting. Some of the research is out on that, 2021. Some in 2022 as well. Talk about anemia, and then I'll talk about functional laboratory interpretation. What I want to stress with that is the origin of the optimal ranges. I actually know the doctor that created these back in the 80s. You know, I'm 65, so you tend to know a lot of people as you get older, and I don't know if you get any wiser, but at any rate, and then we'll talk about some genomics. So let's have a little bit of fun as we go through this. So let's take a look at this first. So laboratory testing. So what is it used for? A lot of this is on the slides, and I do want to thank my wife for helping me put these slides together. Catherine's not here, but she does a great job in making these things look good. So laboratory testing is used for detecting disease, no surprise, guiding treatment, monitoring response to treatment, and monitoring disease progress. What I want to stretch is that, like it says here, it is an imperfect science. You can get a lot of false negatives and a lot of false positives. So I definitely want you to be aware of that on these tests. This is where, and I'll talk about here on this slide about in fact, I'm going to pose the question right now. What if I told you that you already have the tools to know between 70 and 80% of what's going on with the patient with only doing one thing? Does anybody want to guess on that? Anybody want to fire it? You have 80%, 70 to 80% of a chance of knowing the diagnosis in someone by doing one thing. Any takers? Is that no? Anybody on the other side?
Dr. Sodano, somebody is saying listen to them and get a good history.
Absolutely. Thank you. That's it. Now, what I'm talking about with a history, whoever mentioned that, please pat yourself on the back. You get a extra cup of tea on that one, because that is absolutely true. And I'm focused in on the tougher cases. These are the cases that we're all dealing with, and these are the chronic illness patients. And what I have found out through the years, and I do want to share this story with you that will stress this point of, I've done a lot of consults throughout the years, the last 25 years. And a lot of times when you don't know what's going on with someone, go back and redo the history. You can actually reintroduce yourself, you know, as another practitioner in a sort of way. Let me go back and investigate this history on these chronic diseases. I mean, we know if somebody has, say, appendicitis, the white blood cells, 20,000, they get abdominal pain, that's kind of a gimme to get to the hospital, we're fine. But for these cases, it's important to take a look at the history. What I found one of the most important components to this is kind of zipping your mouth and letting the patient either write or listen to them for their story. So please keep that in mind. And also ask this other question that I've learned through the years is, when was the last time you felt yourself? That could be 5 years ago, 10 years ago, 20 years ago, 30, whatever it is, and work from that point. There's a lot of good information that you can garner from that and help guide this patient, because there could be all kinds of trauma that, that could have been elicited. They could have been in went on somewhere where they got a parasitic infection. They were fine up until 16. They went swimming in a pond, got a parasitic infection. There was some kind of abuse or something happened during that time that set the whole adrenals off. So that would be something that I would stress so much to help guide your patients. Now, with regard to laboratory testing, there is an old thing. I don't know if any of y'all, when I was in high school and I had to read Homer, Ulysses. I don't know if anybody knows about that, the Iliad and the Odyssey and so forth. But there's an old term there's a term that's been used called Ulysses Syndrome. Now, Ulysses, he was a warrior during Greek time. And he, after the Trojan War, he couldn't find his way home for like 10 years. And he finally found his way home. So the point here is that if you start going down this rabbit hole of lab test after lab test after lab test, it's going to cost the patient a lot of money, a lot of expense, and you may just end up right where you started, doing the proper history and doing basic laboratory testing. And I do want to add on this 3X genetic testing as well, because you can get the genetic background of the individual patient that marries very nicely with even basic laboratory testing. So hopefully that makes a little sense to you all. Again, you have the tools that are there. Now, with regard to laboratory testing, what is it? Here's it's broken down very simply: diagnostic testing, screening testing, and then routine testing. So that's really the basic purpose of that. Again, remember, last time I'll mention this is that 7th grade science teachers where I mentioned that. History, environmental history. Oh, by the way, I did want to mention this one case, and some of you may have already heard this from me, but I think there's a point to this of repeating this for anybody that's new here. This was about 20 years ago that I had someone ask me to look at their information, look at the console. There was an individual that had migraines, constant, every test, all medications, over and over again, functional medicine testing, and the rudimentary, some genetic testing way back when, because there was MTHFR, that kind of thing, not where it's at right now. And he said, we don't know what's going on. We can't figure this out. And he said, hey, doc, can you take a look at this for me? And I said, sure. So I took the notes, sequestered myself, went over it, and I thought, what's missing here? What was missing is they forgot to ask the patient about family members. Her husband happened to work as one of these, worked for a crop duster, bringing environmental toxins into the house. They also had carpet. So once, and I followed up with this, after about 6 months, they got rid of the carpet. They asked the husband to shower up and leave his, his overall, so forth. Don't bring stuff into the house. Headaches were gone. So sometimes it's a matter of removing things. Now, with the rationale of obtaining laboratory studies, here's the basic rationale. And I also want to point out, please use these notes as a compendium. You'll see as we go through, you can use them as a guide. So hopefully you'll have access to these. You can download them, use them as a compendium. It could be very helpful because I have a lot of tables on here and charts that, to me, and I still use these today, as a matter of fact. So here, establish a diagnosis, guide the course of treatment, rule out diseases, possibilities. You can read a lot of these, detect disease recurrence, assess the effectiveness of treatment. That's why sometimes you want to retest, determine the stage, activity, severity of disease, screen for disease, assess nutrition, and certainly screen for toxins and drugs. And by the way, if your patients are on a lot of medications, go online, look up the adverse side effects, because more than likely when they come into your office, it could be an adverse side effect from the drugs themselves. This is rather interesting. Next, I want to talk about factors affecting laboratory tests. There's a lot of things. There's pre-analytical factors, analytical factors, and post-analytical factors. The pre-analytical factors occurs what happens during the process, before the blood is even processed. The specimens are even processed. It doesn't necessarily have to be blood. It can be stool testing, urine testing. There could be contamination, all kinds of things going on with lab testing. So I want to make sure that you're up to speed on it. And if you get unusual lab test results come back, you want to know what's going on here. Now, so pre-analytical factors, a lot of things that can affect that. One is needle size, the incorrect needle size. If there's more suction in a tube that pulls the blood out, you've all seen that. Excessive heat, prolonged or delayed processing, all of that is going to lead to this one thing, which is the most common pre-analytical factor, is called hemolysis, meaning that the hemoglobin, the red blood cells break down, hemoglobin spills out, all the contents of the red blood cells start to spill out, and it messes up the lab test. So just be aware of that. If you get something strange coming back, even having a tourniquet on for a long time can cause a problem as well, can raise potassium levels and so forth. Next, the analytical factors, that's something that can happen at the lab. There wasn't quality control, so forth. And there's post-analytical factors, which I don't see a whole lot of. That is incorrect data being put in, coming back. fact, I've never seen a post-analytical factor in all these years I've been looking at lab tests, but it is possible, and data entry, things of that nature. So always question if the labs are not consistent with what the patient's symptoms are when they come in with. I've seen quite a bit. This is where you can use this as a compendium. I have this slide and the next 3 or 4 slides. I outline some of the pre-analytical factors that I want you to be aware of. Certainly pregnancy, things are going to change, concentrations, alkaline phosphatase will change. Extreme exercise or exercise are going to change a few analytes, and I have them listed here for you. Pediatric reference ranges, by the way, are not going to be the same for geriatric values. Sometimes they're going to switch a little bit. The optimal ranges that I'll be talking about were done for adults, not pediatrics. A lot of people, other pediatric optimal ranges, well, not really. I don't know anyone that's done them, and if someone does, please let me know. Body weight, if somebody is overweight or obese, they're going to have systemic inflammation. Remember, adipose tissue is essentially an endocrine organ, so it's important to realize that. Posture will change things. Diet certainly is going to change things. If they weren't fasting, you wouldn't believe how many people forget to tell their patients you need to be fasting for these certain analytes, and they forget. Alcohol, oral contraceptives, by the way, are going to increase thyroid binding globulin. Isn't that kind of interesting? So we already know about that. Oral contraceptives can cause blood dysregulation, meaning increased clotting, things like that you're aware of that. Hemolysis, I mentioned. Here are some of the analytes that are going to be increased in hemolysis. That would be lactate dehydrogenases, bilirubin, potassium, AST, ALT, AST, the liver enzymes, and so forth. And if I'm going too fast, someone can tell me to slow down, because sometimes I have a tendency to go real fast. So that's my New Jersey coming out, although I don't live there anymore. Another thing to consider are these biological rhythms. And I have them outlined on the next slide. And I'll wait for the next slide to go over these rather than read through here. Altitude is extremely important when we're looking at hemoglobin levels. If someone is living in, let me see, where was I? Flagstaff, Arizona, you're up 7,000 feet. It's going to be a whole lot different, your hemoglobin value, than it's going to be living where I live. And I live in Delaware, and where there's like, the altitude here is like 10 feet where I live. I'm right on by the Atlantic Ocean. Hydration status, very important to think about hydration status. It is going to change your hemoconcentrate, the blood. If somebody's dehydrated, one way you can tell is their albumin is going to be up. If albumin is up, they're dehydrated, and it's going to change the analytes. It's going to make things look a lot higher than really what they are. So that's kind of a good way to take a look at that. Now, as far as the biological rhythms are concerned, here are the 3 biggies. You have the circadian rhythm. Those are different concentrations over the 24-hour period. A lot of analytes will do that. I'll show you on the next slide. There's ultradian rhythms. Those are analytes that are going to be released in a pulsed fashion. Testosterone is the greatest example of that. Testosterone gets released pulsed throughout the day. So sometimes doing one testosterone, serum testosterone, they're like, man, that thing is really low, or that thing's really high. What's going on? Well, sometimes you need to do multiple and factor that in, of course, with other hormones. There's also infraredian rhythms. Those are analytes that are going to exhibit cycles greater than 24 hours. The most common are during the female cycle. The pituitary gonadotropins, talking about luteinizing hormone, follicle-stimulating hormone, and also estrogen, the estrogens, and also progesterone. We know they go up and down during a normal menstrual cycle. So keep that in mind. So here, this is a chart for you to take a look at. I'll slow down just a little bit. So here is the peak, at the time of peak concentration that the body produces certain things. Let's take cortisol, because you're all familiar with that. Morning cortisol should normally be high, but look at the percent change by the time you hit the evening. That's normal. We know we want cortisol secretion to be extremely low. Look at that. Look at TSH, thyroid stimulating hormone. 2 o'clock in the morning, bless somebody. I think somebody sneezed, bless you. So, or cough. Thyroid stimulating hormone is highest at 2 o'clock in the morning, but there is a 206% change of this analyte throughout the day. The best thing that I can tell you, and I think the most proper thing to do, is what I like to do is this. When I ask the patient to go for a blood draw to even things out, it's usually 8, 9 o'clock in the morning, no more, fasting, and have it done every time you do a blood test at the same time. don't change anything. So you're not going to get this variation in analytes. So you're going to be comparing apples to apples here, which I think is important. The other question comes up, well, my patient is on thyroid medication. There's a lot of folks on thyroid medication, T3, T4, sometimes a combination of both. Here's my suggestion for that scenario. If possible, wait, have your patient do the morning blood draw, the fasting draw, have someone be with them if need be, have them get the morning draw as soon as they can, and then continue with their medications, with their thyroid medications. That works out best. So they haven't had it for like 12 hours. So you get and keep that the same way. That will give you, if you're looking at thyroid condition, Hashimoto's, what's going on, thyroid function. That's kind of the best way to get around that. And hopefully that will make some sense to you. Excuse me. Next thing is that I want you to be aware of is biotin. That's vitamin B7. You all know that. So what do you need to know about this guy? Okay. Biotin is part of, it's part of, believe it or not, this biotin B7, we know it's good for hair and nails, and a lot of people are taking a whole bunch of this stuff, wanting to make sure their hair grows properly and their nails and for youth and all this other stuff. So there's a lot out there, usually in super physiological dosages. The thing you need to know about biotin is that biotin is used as a reagent for these assays for certain analytes you see on the slide. There are two assays. There's a sandwich assay and don't worry about the name of these assays. I just want you to be aware of what to do. But I just want you to be aware of that biotin is used on these assays. The particular sandwich assays are used to assess for thyroid stimulating hormone, HCG. I mean, you could read the list: insulin, ferritin, C-peptide, N-terminal pro-B natriuretic peptide. That's something for the heart. It's a good one to do for if you're suspecting myocardial infarction and congestive heart failure and so forth. It's a real good to take a look at that peptide, by the way, but that's pretty much down the line. Follicle stimulating hormone, luteinizing hormone. It could falsely lower those, and there's like, the lab tests don't match up, particularly thyroid stimulating hormone. The other one is the competitive assay that can falsely elevate these analytes. Your total T4, total T3, free T4, cholesterol, cortisol, estradiol, and testosterone, and vitamin D. So you really don't want these things falsely elevated. So how do you get around all this? Bottom line is, find out if your patient is taking biotin. It may be they may not even know it. So we're going back to getting a good history of what vitamins are they actually taking? What nutraceuticals are they taking? Sometimes you got to sometimes I don't read the label. Oh, I'm doing a multivitamin. If it has a lot of biotin in it, it can mess up those particular lab tests. So we want to avoid that as much as possible. So here's what you do. Find out about it, tell them to stay off it for 3 days. It'll wash out and you're pretty much good to go. And you can rely on the tests, so much better, more accuracy of those tests. That's really the one thing you need to be aware of. This is a real deal out here. So next, I want to talk about nutrients required for proper red blood cell production. So why do we want to know about this? What's so important about this? Well, if you don't have the proper amount of red blood cells, right, what do the red blood cells do? They carry oxygen to the cells for use, and they carry carbon dioxide back. I have another question that hopefully I'm going to throw this one out there. What does oxygen do in the body? It only does one thing, and I want to know if somebody knows what it does. Any takers? Anybody there?
Everybody's very quiet, and I'm watching the chat, and nobody oxidation reactions.
Electron Acceptor.
Who said that? I want to know who said that. Oh, somebody's got their.
I said that. My name is Aygiri.
Oh my gosh! Thank you so much. I want you to think of it that way. Yes, the doctor is exactly right. But I want to break this down a little bit. It is an electron acceptor. So what is an electron? What is oxygen? Right? What electron does it accept? That's extremely important. Well, it's the final acceptor to make ATP. That's it. That's exactly what it does in the body. When you go back and look at these organic acid tests, when you're going through the Krebs cycle, pyruvate dehydrogenase, and all that to make energy, when you look at the oxidative phosphorylation, boom, the last thing to make ATP is that oxygen is in there and accepts that to make ATP to give us energy. So do we need oxidation to give us energy? Yes, absolutely. Do we need antioxidants to calm things down? Yes, it's all about balance. 99, I'm not to say 100% is about balance on when we're looking at, even when we're looking at genetic testing. So you want to make sure that that we have a proper amount of red blood cells, that they're happy, that they're doing their 120 days, and they're pretty much good to go, and they have a really nice, beautiful celebration of life funeral at 120 days, if that makes sense. So the red blood cells, the complete blood count, is one of the most frequently ordered lab tests. And again, it has a good reason. When you look at the CBC, you can look at, is there anemia going on with this patient? Extremely important to know. Is it B12? Is it folic acid? Is it iron deficiency anemia? What's going on? Is there infection going on? Is there cancer? Autoimmune disease? I talked about, and we have slides coming up on the neutrophil lymphocyte ratio. You can pull that off looking at the absolute values that we all get back. And I will mention about the percentages as opposed to the absolute values when you're looking at the differential count. I'll mention that in a few minutes when I review that. So that's why you want to make sure that these red blood cells are happy. There's enough of them. They're doing their job. They're carrying oxygen, removing, what do you call it, carbon dioxide. Red blood cell production. Red blood cells have also been researched out because they are, as long as they're healthy, they actually participate in the health of the cardiovascular system, in particular the endothelial cells, which is extremely important. So that's kind of good information you can get right off of a CBC. Now, vitamin B6, as you see right here, I put the active forms down again. Use this as a compendium. It's the excuse me, pyridoxal 5-phosphate. That's the active form of the vitamin. It's involved in 100 enzymes. You don't need me to read this to you, and all these other decarboxylases, aminotransferases, which, by the way, is in red there. I have it highlighted. Those are the liver enzymes, AST, ALT. They require B6. So from a functional perspective, if those enzymes go low, it may be that the patient is deficient in B6. Without doing a nutritional evaluation, take a look at that. That's a clue if those enzymes go low. I want to talk about this other. Sometimes I just start talking, so hang in there with me on this. If you get, which is not part of this lecture here, but it just happened, I think it's worth mentioning it. If you were to get a patient comes back, well, their liver function seems to be okay, and you get a liver enzyme, one of these, ALT, AST, in isolation that's elevated, and you are scratching your head, and the doctors are scratching their head. What's going on with this? Why is this elevated? I cannot figure this out. Please look for celiac disease. There is a 9% chance that an individual with celiac disease will have an elevated liver enzyme in isolation. In other words, alkaline phosphatase is going to be fine. Bilirubin will be fine. GGT is okay. And it's like, man, I really don't know what's going on. I've seen this over and over again and start to question the patient about that. Now, a vitamin B6 deficiency, it could decrease the synthesis of hemoglobin. And that's something that, we don't want, because hemoglobin, that's the one that has the iron incorporated in it to get the oxygen, deliver it, and the carbon dioxide. So hopefully that makes sense. Next is B12. I always look at B12 and folate together because they kind of marry one another. We're going to look at the methylation profile a little bit later, and this will kind of make sense if you're not familiar with that. But one thing I want you to keep in mind, B12 and folate depletion occurs long before changes are seen in the red blood cell morphology. In other words, those that know about the red blood cell indices, you may not see changes in the mean corpuscular volume. You may not see it get larger with this deficiency. And so it's a good thing to test for this. Also to test for the genetics, which I'm sure you're going to be learning with 3x4. I almost wish I could teach this part of it because I love this so much, but we'll leave that to Dr. Yao, much more of an Dr. Jaffe, much more of an expert than me. Vitamin C. So what is vitamin C? It's a reducing agent. It helps, it's an antioxidant. It helps with glutathione recovery. But vitamin C, it appears to reduce the rate of oxidation on 10-formyl tetrahydrofolate. So what's that? Folic acid. And it keeps the folate pool repleted. That's where this vitamin C, it's important to take a look at. I personal belief, which I don't mind sharing, I think everyone should have a little bit extra vitamin C each day through your fruits and vegetables. And if you need to take a little bit more these days with the immune system, I think it's important because it is a free radical scavenger, and it also helps with glutathione recovery, especially after oxidative stress, because I think we live in a world of oxidative stress. Iron. So let's talk about iron. 70% of the iron is in the hemoglobin. Makes sense, because that's what it's used for, and it binds the oxygen, like I said. But there's another 3.9% found in myoglobin and in the mitochondrial proteins. They're involved in the cytochromes. They're involved. Iron's involved with catalase. Sound familiar? Peroxidase. It's also involved in the electron transport chain. So if someone is deficient in iron, that it could be for a lot of reasons. It could be blood loss that they have, and I'll talk about the anemia of inflammation, which is, I'm sure it's coming up somewhere along here, that the body will stop absorbing iron for a particular reason. I'll wait for that slide. Iron is also needed for the thyroid enzyme, thyroid peroxidase. It is a heme-dependent enzyme. So going back, when you're looking at the CBC, and if you have a low mean corpuscular volume, and if you tack on the iron panel, which I suggest that you do, the iron panel consists of serum ferritin, total iron binding capacity, and serum iron. Those three things. If you need to figure out the percent saturation, which is going to let you know you don't have your iron deficiency anemia, or your iron overload, or hemochromatosis, there's a real simple calculation. If your lab doesn't do it, or if you need to do it, and here it is. If you look at serum ferritin, the value, multiply it times 100, and then divide by the total iron binding capacity, and there you go. There's the calculation. So now you have percent saturation, and if it's high, this person is iron toxic. They're taking too much iron. It could be hemochromatosis, or if it's too low, iron deficiency anemia, which can be caused by a whole bunch of things. So you can see iron is extremely important. Now, the body will sequester iron, will stop it from absorbing when there is inflammation and infection. Reason being, because iron is needed for parasites and microbes, and the body saying, no, we need to kind of get rid of you here. The liver will produce, by the way, a substance called hepsin, which is antibacterial. So the body's trying to work it that way. But chronic infection, you're going to see most of these folks are iron deficient. So getting rid of the infection, repleting the iron, and figuring out what's going on can be extremely important to getting your patient feeling better, because there's a lot of anemia out there. Next, amino acids, they're crucial for just about everything, including the immune system as well. And amino acids are needed for heme production. So it goes without saying, if you have someone that has celiac or malabsorption, even maldigestion, they're not breaking down the protein that they're putting in their belly. It's not breaking down to tripeptides, dipeptides, and amino acids. Therefore, they're not absorbing them properly. Then they get a whole lot of food allergies. Then they get zonulin, and that starts increasing, and then they get a whole bunch of other things going on. So make sure that their digestion is fine. If they need support, it's extremely important, because you want to do it upstream, just like looking at these genetics upstream. So here is the neutrophil to lymphocyte ratio. Okay, so let's take a look at that. So how do you calculate this guy? Let me go to this slide. Let's talk about that. I probably should have put a lab test up here more than I think about it. I try not to put too many things on slides, and I try to make them where people can read them. Thank you to my wife, Catherine, for making me aware of that. I used to load these things down with all kinds of things, and it used to scramble the whole point here. But when you get your laboratory tests back, meaning the CBC, and I'm sure you're going to order a differential on that, you're going to get percentages back of the white blood cells. And then underneath that, you're probably going to get the absolute counts. A word about the percentages. We all learned many years ago, I know I did, that we're looking at the percentages, high neutrophils, low lymphocytes, they mean this, they mean that, functionally here and there. The caveat of looking at percentages, and what you'll see now is not established next to that on the column all the way to the right in the lab test. The caveat is it's the percentage is based on 100. So if you have cells that are like a huge infection, neutrophils are like 70%, everything else is going to be low. So you're going to be reading everything low. They may not be low. This is why you want to take a look at the absolute counts. That gives you an idea. There are no optimal ranges for absolute counts. What I do is take a look at the absolute count that comes back on the reference range, which is over on the right. And I like to just split it right in dead center and call that, Wayne's World optimal range on that to see really what's going on. For this ratio, neutrophil and lymphocyte ratio, just do the math. You'll get numbers back. So just do the ratio. Neutrophil will be a number there, maybe 2.2, lymphocyte maybe 1.0, and just do the math, and then you have a ratio. The thing about this, and nothing is absolute, it's going to reflect the balance between the systemic inflammation and, I'm sorry? Oh, I thought I heard somebody say something. No, okay. Can you all still hear me?
We can.
Let-
We are glued
Yes.
to our seats.
Okay, okay. Yeah, I just want to make sure, sometimes get off, think I'm talking to myself here. I just want to make sure everybody's on board and everybody's happy here. Because that's really what this is about, is making our patients happy and ourselves as well. So here, so looking at this ratio again, you can easily do the math here, right off of a CBC. Isn't this good to know? So this is some information. Again, use this as a compendium. You don't need to memorize this. It's already on these slides. So neutrophils, what are they? A major role in inflammation for microbial infection. They're going to be jacked up. Lymphocytes, that's adaptive immunity. They're going to be part of the natural killer cells, the T cells, and so forth. What generally happens is that there'll be a decrease in lymphocytes, which we will see with COVID, by the way. So it's kind of interesting. So you can get an idea of what's going on with this immune system just by looking at these two absolute counts and figuring out what's going on. And now here's what can happen with chronic infection. This gets a little technical, but I know y'all can handle this. Chronic inflammation, it can stimulate what's known as the release of this immunoregulatory granulocyte from the bone, right? And it can suppress the lymphocyte cells. This stuff can enter into that, and then you'll get this weird ratio. That's how it's picked up in chronic inflammation, immune system dysregulation, and like it says, a high level of circulating neutrophils are associated with the depressive activity of the T lymphocytes and natural killer cells. If natural killer cells are down, cancer could be possibly up, because those are the ones that just like they say, they are natural killer cells, and they're going to go after viruses. They're going to go after cancer cells, and if they're low due to chronic infection, you're going to get a little bit of a window into this. Again, nothing is absolute. If you want more information, I have references that are here, so you can take a look at that. This is 2021, so fairly recent. So what do you need to know about this for this course, just as a starting base? This comes from this reference right here. This was done, we're 2007 notes, and it hasn't changed. So what they say, and I'm just going to read it, we've identified that the normal neutrophil lymphocyte ratio in the adult non-geriatric population somewhere around 0.78 to 3.53. So when you do the math, plug that in there, say, hey, now you know what's going on with that. Now you know a preventive medicine thing to take a look at, which I think is pretty cool. As this relates to COVID, like I talked about, this came out 2020, this article. Here's the reference for that. What they found out, and I'm just going to read it out, patients age 50 or over and having a neutrophil lymphocyte ratio of 3.31 or greater are predicted of developing critical illness. Does this make sense? They are low on natural killer cells here, knowing that, and thus they should have rapid access to intensive care. You can use this as a preventive medicine thing, and you can get it right off the CBC. So hopefully that makes a little bit of sense to you. So the approach to anemia. There's your old comprehensive patient history. Get a really good history, and here's that 7th grade science teacher. Go back and have the patient either write their story or sit down, take the time, and let them talk about it. Probe their brain a little bit. Say, when was the last time you felt yourself? That is a great starting point. It may be childhood trauma, maybe teenage years, something may have happened, because we need to deal with the emotional component. The definition of holistic medicine, meaning that there's a greater amount than the sum of our parts. And I'll just leave it at that. That's a lecture for another time. But I want you to remember that the signs and symptoms of anemia, they are a function of the severity, how bad, how fast the anemia came on, and also the age of the patient, and symptoms can certainly vary. And you'll know that by the lab test here. What is kind of cool on this one is the connection between inflammation, the direct biochemical, and anemia. Aside from the story about the iron that I had told you about, you have inflammation that is going to produce pro-inflammatory cytokines. It is going to decrease, this is erythropoietin from the kidney. So there you don't have erythropoietin, it's not stimulating the bone marrow, and therefore you're getting a low red blood cell count. Pro-inflammatory cytokines are going to shorten the life of the red blood cells. Because there's inflammation, Red blood cells do not have a nucleus. They have a wonderful antioxidant system in them. They need vitamin E on the membrane to keep them nice and healthy. If none of that's there, they're just going to say, you know what, I'm going to cash in around day 90 or something, and they're just going to start to break loose. So that is what's going on here. That's why we want to address the inflammatory cytokines in inflammation. By the way, there is a test to look at those. Avexia offers that. It's the CytoDX test that will look at these cytokines if you need to take a look at those too. I know cytokine genes for those are looked at also on 3x4. So isn't that pretty cool for inflammation? So here's the bottom line. Any disease state with a major inflammatory component may be accompanied by, and usually is by, a hypoproliferative anemia, low production of red blood cells. So please keep that in mind. And iron deficiency, which you already know. Here are some conditions that are associated with reduced erythropoietin response, which is produced by the kidneys. Some of these may be very familiar to you. Chronic and acute bacterial infections, malignancies, renal disease, protein deprivation, malabsorption conditions, celiac disease, I would throw that in there. And of course, your list of endocrine disorders all cause a decrease in erythropoietin, so these patients are anemic. Here's some of the signs and symptoms, very nonspecific. I got headaches all the time. I don't know what's going on. Lightheadedness, hypotension, tachycardia, muscle weakness, vertigo. They're nonspecific, but when you look at the CBC, which is important, these are some of the information when you put, oh wait, there's anemia going on here. You can add that also to the mix. Next, and I think this is the last section. I don't have these slides totally memorized here, but next is chronic blood loss. Which can lead, and it could be microscopic, can lead to iron deficiency anemia. Usually you're going to see that in the gastrointestinal tract. There's going to be mucosal sloughing. They're going to lose iron that way. There could be secondary bowel disease as well going on with that. There could be gynecological problems, a lot of blood loss. So you need to be aware of that. So how do you pick that up? First of all, doing an iron panel, but any presentation coming back in the CBC, low hemoglobin, low RBC, and a low mean corpuscular volume is suspicious for possible blood loss. You can also do a stool analysis as well, but just kind of be aware of that. So those are your flags for that, because you definitely want to address a chronic blood loss. I see a lot of it. This is B12 folate. Now, I know you have the wonderful, the 3x4 genetics that can take a look at all, these wonderful genes to produce the enzymes, which I'll talk about in a few minutes. But I wanted to give you some information on just looking at blood testing, because that's mainly what this is about here. There is your normal range for serum B12, for folate, for serum methylmalonic acid. Methylmalonic acid can show you the functional range of B12, because that's a byproduct. If this thing goes up, that means you need more B12, more than a personalized medicine, more than just looking at their RDA. And this can help you decide what is going on. Usually these things kind of tag together. Homocysteine will also go up, as you can see, so you can get a little bit of an idea if you're doing serum levels, which I suggest you do. Now, speaking of serum levels, I want to talk about this. I have seen this because I guess, when you do something for a living, you analyze blood tests, you tend to see a lot of things over and over again. And I have seen this more than enough times. And this has to do with an increased B12 serum when it comes back when we do a serum test. Excuse me. If the patient is supplementing, you're pretty much good to go, because you know the reason why serum B12 has gone elevated, because they're telling you they're supplementing. So naturally it's going to go up in the serum. If they are not, and again, you can have two things going on at the same time. If serum B12 is elevated, suspicion goes up for a couple of conditions. One is the myeloproliferative condition, which is possibly environmentally induced, because it can cause an increase in red blood cells, increase in platelets, and all kinds of things. They think there's a genetic component involved in here, and it could also be environmental toxin component. Also, liver disease, malignancies, renal disease, and malnutrition can all cause an increase in serum B12. What happens is that this, I'll just go over here and outline it. This transcobalamin, that is a vitamin B12 carrier protein, will become increased in these diseases, liver dysfunction. When liver becomes damaged, it starts spitting all kinds of stuff out. That's when you get the The aminotransferases run high, GGT run high, because the cells start breaking over and these enzymes just spill over into the blood, and there you go, they're high. Same thing can happen with transcobalamin, will go high, but that will cause an increase in the level of B12. Some conditions involved in that, autoimmune conditions, leukemia can be involved in this as well. So this is definitely a red flag. There's a reference down here for you to take a look at through 2021, just in case that comes back. Now you know what the potential is. Most often it's due to supplementation. Okay, I needed to get a sip of water. Hang on a second, please. Oh, Three Stooges. Okay, so next, let's talk about these functional ranges, optimal ranges, functional medicine interpretation of patterns. Once again, use this for a compendium. But I want to start out by honoring the person that I learned this from and the first investigator for this. I would never teach this without mentioning Dr. Harry Eidner's name. He's a friend, he's a colleague here in the States, so I want to thank him for allowing me to share this. And he taught me this, so this is way back when, so that's where this comes from. I'm not all that smart, by the way. So Dr. Eidner, what they were doing back in the 80s, this is kind of a really neat thing. What he told me, I want to relate his story. He was telling me that back in the 80s, before all the lab testing, I mean, we have so much, but think of the 80s and think now, like 40 years ago, what they were trying to do and what he was trying to do was known as the chemical biopsy. He was trying to look at laboratory tests and investigate with his investigators and colleagues, these are all a bunch of PhD folks, and saying, is there something going on? Do we need to look at these ranges and look for biochemical changes that would lead us to cancer prevention? That's where he told me they started out. He actually sent me a lot of his research, which is really cool. So they were looking for patterns on the analytes of particular conditions. Now, as it says here, they looked at over 10,000 cases. They also did this to give this a little bit of power behind it. They did electrophoresis. They did atomic absorption spectroscopy on this. They looked at hormones. They looked at hematological studies, the physical exam, urinalysis, stool analysis, hair analysis, and then came up with these optimal biochemical ranges. Where are things functioning properly? That's why the ranges appear to be a little bit tighter. And what I have seen over the last maybe 10 years, and when I look at these other lab companies, they're actually tightening up their ranges that Dr. Eidner and company did way back 40 years ago, which is kind of cool. And I'm glad to see them do that. Looking at liver enzyme ranges have gotten a little bit tighter. Some of you may have noticed that. But what they said is that these conditions are not absolute. You got to go back to the patient history, and you don't want to make a diagnosis just based on the lab test, but they can give you a good window into that. Now, what I have outlined on this slide and the next, I think it's 3 or 4 slides. I kind of lost count here. For you to get some kind of an idea on what he did, this is a little window on what they did, the researchers did. They looked at CBC and the comprehensive metabolic panel. Basically, those were the 2 main tests. Of course, they looked for other things as well, but those are the 2 tests that you really can get a lot of good information on conditions. Is this person in acidosis? You can easily get the anion gap off of a comprehensive metabolic panel. Those analytes will be increased, meaning chloride, sodium will be increased. You'll see a decrease in CO2 will be decreased as well. That's how they come up with that. Adrenal function. Wouldn't it great to have a little window into adrenal function where you see potassium go up? But we know if the adrenals aren't working well, there's going to be a decreased cortisol, decreased DHEA, decreased aldosterone, because all that stuff's made there. And aldosterone does what? That's involved in sodium and potassium back and forth going on to. So that's why if that goes low, that's all that balance is going to be messed up as well. Potential for food allergies, looking at the eosinophil count and basophil count. Looking at total serum immunoglobulins. If they go down, again, the immunoglobulins are the ones that latch onto things. Right? If that starts to go down, hey, is there some food allergies, some environmental toxins? Those are the questions as you look at that. Look at the test when it comes back. Say, oh, that's where that could give me a clue on what's going on. For anemia, hopefully you always do an iron panel with this. I wouldn't leave home without that, and also a thyroid panel. I just think it's these days, if you're ordering blood tests, that would be the basics to look at. Anemia, iron deficiency anemia, you'll see this analyte, total iron binding capacity. These will go high, these will go low. I think you got that. They figured out that there's a possible need for hydrochloric acid. Now, why would that be functionally? Well, as it turns out, hydrochloric acid it helps to chelate the iron and also, vitamin C to help it for absorption. So if somebody is low HCl, or as somebody being older could be chlorhydric, which can happen as they get older, where there's something going on with intrinsic factors, so forth. They're going to have a tough time absorbing iron. So you need to get something that is more bioavailable to that person. So, please be aware of that. We talked about anemia being vitamin B6 deficiency. You already know about that. Analytes high, analytes low. I don't need to read these to you, but I think you're getting the drift of what's going on here. This is all outlined. Again, use this please as a compendium. Other things that they came up with, and they came up with a lot more than this. I just wanted to share this information with you because I think it is extremely important to know about that. By the way, inflammation for the gut, this kind of interesting, digestive inflammation. I think it's interesting that protein certainly goes low. Does that make sense? Albumin going low, hematocrit going low. You're just not absorbing these things properly. There's inflammation. So those are what they have figured out looking at those 10,000 plus cases, gastritis, gout. Okay, I think you got it. I'm not going to go over each one, but you get a good idea. This information's here. And parasites, the eosinophils. But what I want to get to, zinc deficiency. Zinc is seen as a cofactor for alkaline phosphatase. What's kind of cool is this, is that without having to memorize all of this stuff, Avexi has what's known as their functional health report, that there are panels that are created, and they're not profiles, panels, that if you order particular panels through Avexia, they will put this in a computer program and take a look at these optimal ranges, standard ranges, and alarm ranges and let you know pretty much what's going on, and also do all of that work for you that you saw on the previous slides and then some. So I just wanted to share that with you. Now we're getting near the end. How much time? Not too bad. We've been into this close to about an hour. Hopefully you all are still awake. I try to not go more than an hour because everybody sometimes falls asleep at around 50 minutes. Anyway, hopefully you're all hanging on there. Let's talk about genomics. You have a wonderful teachers going to talk about genomics at 3x4. Dr. Jaffe, super, written some books that I know of, and I do need to get one of your books, Doc. Anyway, so let's talk about rudimentary genomics. I'm sure they're going to expand a little bit more on it, but let's look at it from this perspective. I think this is a good starting point as you're beginning this course, the master class. So let me just read it. Genomics. It is a term that describes the study of all of a person's genes. In other words, their genome and the interactions between those genes with each other and with a person's environment. May want to read that again to yourself when you get a chance, because that's pretty powerful. Now, genomics includes like the scientific study of complex diseases, heart disease, asthma, diabetes, cancer, because these diseases are typically caused more by a combination of genetic and environmental factors than individual genes. Isn't it cool that the 3x4, when you get into that, they are looking at a lot of combinations here and going to give you some good feedback. So please keep that in mind when you're looking at that. Next are these, the infamous SNPs, these single nucleotide polymorphisms. So essentially, what are they? You should remember the DNA base pairs: adenine, thymine, guanine, cytosine, all those pairs. There's the 4 letters of the alphabet, which are referred to. SNPs are genetic variations. I want you to think of SNPs as not good and not bad, as I was online with Dr. Jaffe the other day, and she put that very, very eloquently, as because we are evolving with our environment, things will change. Sometimes these things can be normal, and we are also changing our genetic background. So a SNP can be sometimes beneficial and not beneficial. But here's the situation, that the allele, which you need to know about, there are 2 alleles for each gene, one from each parent. When you get 3x4 information back, you'll see maybe an AA or an AG somewhere letting you know what's going on. Now, a SNP can present as either or both alleles or none at all, which would be heterozygous, homozygous, or the wild type, respectively. What's important to know is that the expression of the genotype or the makeup is referred to as the phenotype. What are we looking like? Eye color, hair color, things of that nature, our body type. Male, female, that's all phenotypic expression. What is the DNA telling us? I've learned years ago that the DNA has all the information to do everything, and most of the stuff is turned off for the most part. When we talk about epigenetics, which I'm not doing here, that is food, environment, emotions, toxin, things like that can turn things on and off with turning genes on, turning genes off. Some good stuff and some not so good stuff. One of these days I'd like to do a DNA methylation class. Here's back to these SNPs, right? So SNPs do not directly cause disease, as it says here, but they can affect enzyme function. What's neat about this is that they can be modified by diet, nutrients, environmental toxins. Removing toxins can help these things work more efficiently. Multiple SNPs in a gene, right, can increase the odds for an abnormal phenotypic expression. In other words, you're not making enough folic acid, as an example. Now, I have two genes here listed, MTHFR. This is the C677T plus the A129-8C. Those are two genes for methylation. If you have one, maybe something can be a little bit off, but if you get two, then you get three, and then you get four, it could be a problem with the MTHFR. So essentially, SNPs in multiple genes for enzymes in a common pathway process may be necessary to affect metabolism. In other words, one off, as we would call it here, it may not really do a whole lot of anything. So you need to look at the phenotypic expression, what's going on with the patient, and look at pathways, which 3x4 actually does. So I want to take a look at methylation pathway. So let's take a look. Now, why is it so important? Methylation, by the way, and not on here, but I want to mention methylation is important for turning genes on and genes off. If you get an opportunity, look up DNA methylation. Keeping methylation properties is very important for the expression of the gene itself. What I'm talking about here in this pathway is right here. Methylation pathway being important to make DNA, RNA, proteins, neurotransmitters, and phospholipid membranes, and whatever else they do, which is a whole bunch of things, peptides and so forth. So when you look at this methylation cycle, which is here, starts with methionine. Here is what I call the universal donor, SAM, S-adenosylmethionine, which is right here. SAM is very closely regulated, by the way. The liver has a lot to do with this. It's usually other things not working well that messes this up. In other words, you're not removing S-adenosyl homocysteine properly. And that leads to, it could be more this area getting jammed up, needing some B6, GGT, that there's a problem with that enzyme. This is the folate cycle over here, not working really well. So it could be a combination of things. What I have listed here are the enzymes, MTHFR, and you can have them all listed here. I'm not going to read them all to you. There's a betaine homocysteine methyltransferase. This is the short route to take homocysteine, recycle it to go back to methionine to do what? To get a methyl donor to do all of this. That's the point. Homocysteine can also go down this pathway for sulfuration. What is that? Adding a sulfur to it to make glutathione. So this has many areas to be jammed up. This short cycle, by the way, or short arc, that uses choline. It can use proper DHA, the essential fatty acid. So there's a lot of backup systems. So in other words, if you have one SNP that's out, right, it may not affect this thing because there's a whole bunch of backup. That's why you want to look at things in sequence. But what I do want to talk to you about is functional testing. Functional testing, remove the genetic SNPs and what they do, what they don't do. When you get a functional test back, it's going to tell you in the plasma, for an example, a plasma methylation profile, how much homocysteine do you have? How much N-adenosyl methionine do you have? How much N-adenosyl homocysteine do you have? How much cystathione do you have? Isn't that so? You can you can put that into this equation and said, where do I need to put B6? I may need to put more choline, more DHEA. I may need to use 5-methyl tetrahydrofolate because this enzyme is not working properly. Where 5,10-methylene tetrahydrofolate is not going to go to the MTHFR because I need to help with B12 for this entire pathway. So to sum all that up, and I'll slow down, this is an example of functional testing. You get these analytes back from this pathway on this particular methylation profile. It takes a look at the plasma, methionine, cysteine, S-adenosyl methionine, S-adenosyl homocysteine. It looks at homocysteine and cystathione, and it puts it out into this number, reference ranges, and what is going on here. Here you see SAM is right here and right here, because I like to point to that, because that's the big dude that is that methylation donor. right, to do all of these like we had talked about. So this will let you know where are things being jammed up if these things are all out of whack here in this case. Right? If there was more cystathione in this case, see, it should be greater, it should be less than 0.5 micromoles per DL. It's higher. So is there a problem with B6 doing this conversion where it's not going to cysteine? And by the way, glutathione should be down here too, so forgive me on that. So that gives you an idea. Also, I'm sorry. This SAM, they call it the Samsara ratio, which is the ratio of S-adenosylmethionine and S-adenylyl homocysteine. I really like this ratio because it gives you a little window into hypo or hypermethylation. If this thing is off, and usually it has to do with SAW, right, S-adenylyl homocysteine, not going down this pathway, something's jamming up here. That's usually what happens. Usually it's not an overload of SAM, but by the way, in my experience, unless you're not getting enough methionine. There is malabsorption, things like that. So you kind of get a good window, but this gives you a good window into that. I know this is technical, but I know you all can handle this. You're all a nutritionist. I think you got this. So I want to look at, we're getting near the tail end here. So let's have a little fun looking at some genes and some enzymes that they produce. So as it turns out, the COMP gene, catechol-methyltransferases, produces the COMP enzyme. Isn't that fascinating? So it produces catechol-methyltransferase. So what's the deal about this enzyme as we go through this? Well, this enzyme is involved in inactivation of the catecholamines, meaning dopamine, epinephrine, norepinephrine, but also the catechol estrogens. It's important to get estrogens to detoxify, and this goes definitely to phase 2 detoxification, which is what I'll show you, or else there could be a backup in the system as we look at the estrogen pool, and I'll talk about that on the incoming slide. It'll make more sense. But this gets a little technical, but I do want to read this to you. So research has demonstrated that a common variance in the COMP locus results in the replacement of the amino acid valine with methionine at position 158 in the enzyme. And here's the deal, which causes a dramatic reduction in the enzyme's ability to metabolize these neurotransmitters and catecholamine esterases. In other words, there could be a backup in the system. Don't worry about the technical. I brought this up because there's a slide that I have on valine and methionine for you to take a look at. So let's dig a little bit deeper into this one. So like I said, the COMP, it has a role in metabolizing estrogens. The thing that I want to think about it this way with metabolizing it. Why do you want to metabolize estrogens? Well, if the ovaries keep producing estradiol, after it touches the receptors on the cell and it's done doing its job, telling the DNA what to do, right, for cell proliferation or whatever, it needs to get out of the body, or else, by the end of the year, you're going to be nothing but one big ball of estrogen. Right? It has to be detoxified for balance. A lot of things need to be detoxified to get the balanced amount of in this case, hormones, but also neurotransmitters. So I want you to think of it that way. That's where this balance comes in proper detoxification. Now, the catechol estrogens, they're formed during this process of metabolism of estrogens, such as the estrogen estradiol. Now, there's no such thing as estrogen. It's actually a combination of the three, estradiol, estriol, and estrone, and by the way. Now, the catechol estrogen inactivation, removing these things through phase 2 detoxification, it's going to decrease the cancer-causing potential of certain metabolites, the 2-hydroxy estrogens and the 4-hydroxy estrogens, because they can be pro-carcinogenic. What they do is they will create these, they can be further oxidized in the body. So if somebody's under oxidative stress, to what's known as estrogen-O-quinones. Just know that these quinones are kind of bad dudes and can go after the DNA and are related to breast cancer, where you want things to go down the more protective pathway, where COMP can create more of the 2-methoxy estradiol, and that's what you want there. This is for your reference material here. I'm not going to read the good and the bad. You can take a look at this, and this graph here sums this up, is what I'm talking about. Think of phase 1, phase 2 estrogen metabolism. First of all, the hormone it's done its job. It's touched the receptor. It's told the uterine cells to proliferate, as an example. In this case, we'll talk about the cycle. So it says, okay, we need more uterine cells. Go for it. Estradiol, go ahead and go for it. Create more. But you've done your job. Now you need to get out of the body. You don't want to have more estrogen, because now you have more cell proliferation. And where do you get? Cancer, more cell proliferation, which is precancerous things. So you want to go through phase one. Phase one means essentially that the body is recognizing this thing as a toxin and we need to remove it. We need to get it out. There's too much of it. So that's phase one. And that's done mainly by the cytochromes, the P450s. All right, here you can see 1A1. You don't need to know all these things, but just for complete sake, the cytochrome 1B1, cytochrome 3A4, they will recognize the as these estrogens after they've been used to, okay, we need to get rid of you, we need to metabolize you. The question becomes, where are you going to get metabolized? As you can see in the green, you want more of this protective pathway. And there's the comp enzyme right there, and methylation. This particular person was low on methylation. I would do a methylation profile on this person based on this thing going low. This is a Dutch test example, by the way, for the dried urine test, which has a lot of merits to it, and I do like this test. Here, you can see here, if it went down this pathway in the red, here's the quinones, they're very active, damaging the DNA. So we want to pitch things more towards this way. Now, there could be SNPs on these enzymes as well, which 3x4 is going to let you know about those. So you get an idea if you want to go more the protective pathway, where we may need to use more cruciferous vegetables, DIM, and indole-3 carbinol, and so forth, things that you're familiar with. Hopefully that makes sense. This is a way back slide that I really like. Just to put this whole thing, into context here, because there's a lot of problems that you know of, estrogen metabolism problems, hormonal metabolism problems. I want you to think of it this way. Here is what I just talked about. There's a cell, there's the receptors, the alpha and beta receptors for the estrogen receptors. The ovaries are making estrogen, adrenals are, backup system as well. They're doing it, making reproductive hormones. The liver makes sex hormone binding globulin. It says, okay, how many of you are going to be free to do your job? If you're bound, you're not going to be active. So you get that. But once it's bound, it goes through the phase one detoxification. There's a cytochrome P450 hydroxylases. There they are. They're listed out. So it's okay. We recognize you as a toxin, which once something gets recognized as a toxin, it can become more toxic if it does not go through phase two detoxification. And usually it does. Here are the quinones. That's why you want to get these quinones to be going in another direction for methylation, sulfuration, and glucuronidation to get rid of these things and excrete them properly. That's where the mechanism of excretion is proper bowel movement. You may do some functional medicine testing for stool analysis and you see glucuronidase through the roof. Well, what can happen is bacteria can make, if they're dysbiosis, can make a lot of the enzyme beta-glucuronidase. It will deconjugate these conjugated estrogens and go back to enterohepatic recirculation, go through the gut, back to the liver, and put these things back in. And all of a sudden, you got symptoms of high estrogen. You need to think of estrogen as the estrogen pool, the free estrogens, sex hormone binding globulin, the estrogen metabolites. A lot of these can go back and re-hit these receptors. So it's important to take a look at that as well. And proper detoxification, fiber, basic things. Make sure someone's going to the bathroom, they're drinking enough water, things like that. Hopefully that makes sense. Back to this comp enzyme. Just so you know, if you get the valine, methionine, I'm not going to do this one. Let's go here. This is some of the things so you can really get an idea of what's going on when you get the genes and the alleles back. Here, if you have the valine, high enzymatic activity, but it also can affect things other ways that may not be even with the wild type. So it's kind of interesting. Dopamine levels may be a little bit low. This person here, pain medication, may need a higher dose. I have had patients that have reported getting a shot of morphine and it doesn't do anything to them. I've had that happen, and that has to do with genetics. Enzyme activity, if it's very low. Knowing this information allows you to kind of plug in and say, what do I need to help do with this patient? If you have the MET, maybe they need some more SAM, methyl donors, magnesium, some DIM to help them out. That's where this ties in with the functional testing. Now, last part, we're done, almost done here. Nutrigenomics, nutrigenetics. You'll be hearing this until you're, it kind of comes out your ears, and which is a good thing. So nutritional genomics, okay, that's the interaction, as you already know this, the bioactive components of food and genome. We are associated with our environment. We are connected to our environment. If someone doesn't believe that, I want you to give me a call and I want to have a discussion with you if you don't believe that. That's why I don't like any form or shape of polluting the environment. I look at it as polluting our own body, because somehow we're going to be sucking this stuff up and it can change our dynamics and cause illness. Now, nutrigenomics, right? That's the ability of bioactive food to influence the gene, looking at the genetic background, right? Nutrigenetics is looking at the genetic variations and how they're going to respond to that food. So, and sometimes you can get changes in absorption and metabolism based on your nutrigenetic background. And that's where looking at the 3x4 genetics, say, okay, this person's not metabolizing this properly. Can we tweak the food components? Can we tweak their exercise, their diet, which is all provided that, and help kind of write the course or look at this genetic background and help that individual. So to sum it up, here we go. You got your nutrigenetics, nutrigenomics. Again, individual phenotype, what's the expression of all this stuff? And we hope that it certainly is help. Nutrients, genes, you got it. This last part, this is the absolute last slide. I already know that one. I just want to throw this one up there for you, just something to kind of dabble on. This is an older one. This was done in 2004, but I think it's worth going back and giving credit to those that really were taking a look at this many years ago. These are the tenets of nutrigenomics way back when, and we're looking almost 20 years ago. And so there's a lot of good research on this. Common dietary chemicals can act on the human genome. Hey, how about that? Can directly or indirectly alter gene expression. Yeah, we know that. Again, these are the tenets. Under certain circumstances, some individuals' diet can be a serious risk factor for a number of diseases. Wow, we know that. Some diet-regulated genes, they're likely to play a role in the onset, incident, progression, and/or severity of chronic diseases. That's where we're looking at the genetics. I don't know where that's coming in there, those lines. Anyway, I don't know how that's getting in there. Did somebody just cross that out? No? I have no idea how those lines got in there.
Apologies, we have had that happen. So, and we also don't know how it comes.
Oh, I think it's pretty cool, actually. Maybe it'll draw a picture for us. The degree to which diet influences the balance between health, looking at the genetic makeup, looking at your genetic background. That's what you're doing, again, with the 3x4, which I said like 20 times now, and individual biochemical individuality. So it can certainly help out. Anybody have any questions? Because I'm all done here. Thank you for your time. And by the way, thank you for the opportunity. If you can't tell, I love sharing this stuff and love learning it, and will continue to learn, because I don't know everything, but thank you.
Dr. Sodano, as always, you had me on the edge of my seat, riveting, really interesting and exciting information. And I'm sure most of our attendees are going to be going back and re-listening and printing out your slides. So thank you for your wealth of information and knowledge. I'm going to ask Annalie to step in now because she's just going to share the tool that pairs your information with our test. And then we'll get on to questions, if that's okay.
Yep, I'm good. I can sip my tea, which has gotten cold by now. It's okay. Thank you.
Dr. Sodano, can you just stop your share? Then I'll quickly share my screen.
Stop my share. Okay. All right. You good?
Yes, thank you.
Okay. You're quite welcome.
Right. So I hope you can all hear me okay. Nikki, is it okay?
Perfect.
Okay, so you all know, if you are part of our network, that we have a clinical guide, and in our clinical guide, you will find a lot of information about genes, and at the bottom, we've got functional testing for each pathway, and if you click on there, it will give you a lot of information about the types of tests you can do, so that is there. But we have also gone further, and we've actually put a tool together, which is for our masterclass, together with Vivexia a while ago. So what we did is for each and every pathway, this is alphabetical, starting with blood clotting, blood pressure, and bone health, cholesterol, all our pathway, every single one of them. We have panels that we put together, and this will be in your elite access. So you'll be able to see if somebody
Annalie, your sound has gone funny.
Blood clotting genetically. These are the panels you can do. To go to educating myself. Sorry,
Ellie. Won't you just backtrack a minute or so, just saying that if you, we're already there, but we lost you in your tool. Just saying how you can use the tool with the 3x4.
Okay, so you will be able to click on the links and it will take you to the place where the panels are explained. Okay, so this is in the community platform, which is here. So if you go into your community, there's a link here called Educating Myself. If you click on that, it will take you to the webinar. Sorry, I think it's very slow to do both on Zoom and our platform. But anyway, if you go there.
Already in Educating Myself, so you can just scroll.
Okay, then you go here and educational webinars, you can click on here. You can see all the webinars we've done so far. And there's a whole section which we will fill up now with Masterclass series. If you click on that one, you will see all the tools we've already loaded for you, the blood clotting the panels. This is the tool you can use to see all the tests we recommend for each of our pathways. There's a lab testing article. That's great as well. So here, sorry, and as well as the slides are here. So that's all I need to say.
Awesome, thank you, Annalie. So perhaps we can go back to Dr. Sodano, and hopefully whoever's drawing on our screen again will stop. Dr. Sodano, we do have a couple of questions, if you wouldn't mind answering them for us.
Okay.
The first question is, what are your thoughts regarding food sensitivity testing, including MRT?
Okay. I think food sensitivity testing is great. Okay, so I'll start with that. I don't have enough experience with the MRT testing, quite frankly. That's the mediator release test, looking at what is it? All I'm trying to think. Leukotrienes on access my memory here, looking at histamine and so forth. I think it has value. I like food sensitivity testing for this reason, but there are many. But one of the top ones is if I want to initially, when I'm working with a patient, and I know that they have some dysbiosis, I want to take the pressure off of the immune system initially. That information is going to allow me to get the pressure immediately and say, look, you need to stay away from these foods. You're not going to fix leaky gut in a day, kind of thing. Or if there is an increase in tight junctions as we're doing gut restoration. So it allows me to take the pressure off of the immune system, to recognize those foods to stay away from. If I need to go further, I do like a combination of the IgE, IgG, and IgG4. Because the IgG4 is a blocker of the IgE, meaning that you really need to stay away from them. They're causing an immune reaction, and we do want to take the pressure off of the immune system, because how many things are autoimmune, immune dysregulation, gut, and so forth. So yeah, do I like to test? Yeah, absolutely. Be aware though, with this testing, that if the patient, if you're looking at strictly IgG, since it's not a fixed reaction, if the patient was not ingesting these foods, they may not be reacting to it. And also the other thing is there may be foods on there. I don't know why I'm reacting to this food or why it showed up. There's a lot of cross reactivity. So please be aware of that. But initially, great test.
Awesome. The next question we have is, if someone is a slow detoxifier, is 3 days sufficient for biotin to clear before testing?
The short answer is I don't have a clue. Because I'm not sure the half-life of biotin, but here's, when I did the slides, they said 2 days, so I dumped in 3, quite frankly, okay? there was like, when I did the research on it, they said, and I don't know how slow the person is detoxifying, but they said 2 days, you're pretty good. And, you know, in Wayne's world, I'm thinking, you know what, if 2 is good, I'm going for 3 here, just to be on the safe side. So
a little bit of a buffer that you've put in there.
Yeah, so I put in the buffer.
We love Wayne's world.
Yeah. So that I took a little liberty on that one.
Okay, awesome. And then the other question that we have is, would we need to still test serum B12 with methylmalonic acid? Because this practitioner says she typically only does MMA, but from your slides, it seems like it may be beneficial to do both.
Well, yeah, I mean, again, it's going to depend on the patient history. I mean, the MMA is going to give you a really good idea of the functional use of B12, which that practitioner already knows. based on the patient history, if there's a lot of other things going on, supplementation, you're really not sure, I think it may not be a bad idea to go ahead and test for serum B12 and also serum folic acid, looking at that whole methylation pathway. So again, it's going to be a clinical decision. If I had to do one or the other, I'm going for the MMA, quite frankly, looking at the functional.
Awesome.
But
Awesome.
if I know there's a problem going on with the liver, there's a potential for myeloproliferative diseases, I'm going after this thing on the serum.
Awesome. And then just a comment from one of our practitioners that the lab reference books that she uses, obviously your doctor that you referred to, doesn't provide lab ranges based on altitude. And my question is, does your book?
Yes, it does.
Yay.
My book, can I be self-serving for a moment?
You may.
Is that okay? Which I do not like to do. But I do want to talk about this part of the book, so that people that are interested. The appendix, as you know, is huge in the back. It looks at over 200 analytes and says, okay, what would cause this analyte to go up, go down? What kind of drug would do that? What kind of non-medical condition would do that as well? So it looks at all of those. There is a section in the book that looks at anemia. Now you see how I stressed anemia on here. If you were to get that book, there are 4 or 5 pages of an algorithm that will look at hemoglobin, red blood cells, look at the mean corpuscular volumes and everything, and find out, is this thalassemia? Is this thyroid dysfunction? Is this iron deficiency? You could just do the math on it and kind of figure this out on where you're going with this patient. So it does have that in there. A bunch of other things too, but anyway.
Awesome. I'm going to ask you to remind our audience right at the end what the name of the book is. One last question is, would you consider Evusheld in these high G to L patients? I'm not 100% sure.
Can you repeat that?
Yes. Would you consider Evusheld in these high G/L patients? Not sure. Dawn, if you want to elaborate quickly in the
yeah,
chat,
let me ask the question. I was writing very fast. Granulocyte to lymphocyte ratio associated with higher mortality from infection. In the United States, we have available a injectable antibody that lasts for 6 months. It's called Evusheld, and it has good properties against COVID-19. So I was wondering if that ratio might be considered a form of immunosuppression. Locally, at the moment, we have resources in California to give this injection to people who are at high risk for serious COVID, and there's a non-specific immunodeficient code. So if giving antibodies, think of it analogous to gamma globulin. So if giving antibodies would help with this situation, if you had an opinion about that.
First of all, could you repeat the name of that medicine?
Evusheld.
I'm writing this down. I'm not familiar with that.
It's not gotten enough press, but it is actually looks good from a functional medicine standpoint.
Yeah, go figure with this, because I'm all in on whatever someone needs to do with regard to this COVID and looking at certainly the neutrophil lymphocyte ratio. Honestly, Doc, I don't have an answer. I don't have an opinion. I would have to study that and kind of look at that and then figure out an answer to that. I don't have one offhand.
Thank you.
Thank you. Thank you for bringing that up, by the way. I got a little bit of homework to do.
Awesome.
Me too.
This was so amazing. And as always, our practitioners who join us, also so amazing. Thank you for your questions, your comments. I think a lot of the other questions that were posted in the chat have been addressed. One other one, the ratio of ferritin. Can you give us that equation again quickly?
Oh, is that looking at the percent saturation?
Yes.
I think that's what it is. Sure.
Yeah.
Okay, it is serum iron times 100 divided by the total iron binding capacity will give you the percent saturation.
Okay, perfect. I've actually popped it in to the chat if anybody is looking for it. So I hope I didn't make a typo.
Yeah, no, that's
Dr.
it. I see it.
Okay, awesome. Dr. Sodano, this was amazing. Annalie, I'm not sure if you want to jump on and just say our goodbyes. Anything else you want to share quickly?
No, I think this is fantastic, Dr. Sodano, as always.
Thank you
My
so
internet
much.
seems to.
Yeah, here's the,
glitch.
you guys
There's the
wanted
book.
this. There, that's, I
Awesome.
don't know if you can see that or not, probably can't, but
And the best place I believe to get it is on Amazon.
yeah, if you needed to do that. But again, this was an honor, and you can't tell, I love doing this stuff. I learned something.
We can tell.
I'm going to add to this as well, and thank you all so much for asking me. An honor.
We always love having you.
Thank you very much.
Thank
and thanks
you.
for everybody who hung on till the end.
Yes, thank
Yeah.
you.
See you
And
next
see you
week.
next Friday.
Yeah, sounds good. Good luck
Thanks
with
very
the
much.
the master class. Hopefully this got it kicked off, a good kickoff for everyone. God bless everyone. Thank you.
Thanks
Thank
so
you.
much.
Bye.
Bye-bye.